Hey, Bio2 students, Mr. Yance again. Kind of going to pick up where we left off the other day. I want to start with a little video. May not play all of it, but this is kind of a complex topic. What is restriction enzymes? What does this diagram mean? I went over a little bit yesterday in notes, but I kind of want to start with it and follow up and maybe explain a little bit more today so that we understand exactly what's going on here. It's if you go back to the definitions that we used with the introduction of notes, I talked about what is biotechnology, what is genetic engineering, what are we using it for, and the key is to make useful products. So one thing that we do use it for is to make human insulin. So I kind of want to pick up where we left off with that. And what this picture represents. So I'm going to play a short little video, and then we'll kind of come back to that. Okay?
All right, so in this video, we're going to be talking about something known as restriction enzymes. Restriction enzymes. Now, what are restriction enzymes? Well, let's go through an example, and hopefully that'll help answer that question. So let's imagine that we've got a bacteria. And it's just floating around, it's doing its thing, and out of nowhere comes this virus. And the virus is just kind of upset today, so it decides that it wants to infect this bacteria. So what the virus does is it goes over to the bacteria, and it basically attaches to it, and it injects viral DNA into the bacteria. So this is the viral DNA, and I'll just label that here, so viral DNA. And the bacteria, being a living creature, also has its own DNA. So this right here in blue is the bacterial DNA, and I'll just label that here. So bacterial DNA. So basically the virus infected the bacteria, but the bacteria wants to figure out some way to destroy the viral DNA. And one way that it can do that is by labeling its own DNA. So let's imagine that the bacteria labels its DNA with these purple dots. All right. So what these purple dots actually are is a methyl group. So we'll say that these purple dots are methyl groups. In order for the bacterial DNA to be methylated, there's an enzyme known as methylase. So methylase is an enzyme that basically floats around, and as bacterial DNA is synthesized, this enzyme methylase goes around and methylates bacterial DNA. Now, what this basically does is it allows the bacteria to recognize its own DNA and recognize any DNA that's not methylated as foreign DNA. So what we now have is another enzyme that's floating around in the cytoplasm of the bacteria, and that enzyme is known as a restriction enzyme. So this restriction enzyme. So this restriction enzyme is kind of floating around, it's doing its thing, and it recognizes the methylated bacterial DNA, but then it sees this foreign unmethylated DNA and it goes and destroys it. So this is basically a way for bacteria to protect itself from being infected by viruses, and it basically does so by methylating its own DNA and destroying any other foreign DNA that is unmethylated. So the reason that this is called a restriction enzyme is because researchers were noticing that certain bacteria were actually they were resistant to being infected by viruses, so they were basically restricting viral growth. Thus, when they figured out what enzyme was causing that, they basically called it a restriction enzyme because it restricted the growth of viruses. Now, in order for the restriction enzyme to work, it needs to recognize a specific sequence, a specific bacterial sequence. So let's go ahead and write out an example sequence. So imagine that if we zoom down to the nucleotide sequence of the bacterial DNA, let's imagine that maybe the sequence was GAATTC. And of course, if this was one strand, then the sister strand would be GAATTC. Now, I don't know if you noticed, but there's something unique about this specific sequence. So if you read this top line from left to right, GAATTC, and you read this bottom strand from right to left, GAATTC, they are basically identical. And this is known as a palindromic sequence. So palindromic sequence. Now, basically what a palindromic sequence is exactly this, and it's what restriction enzymes recognize. So let's imagine that we have a restriction enzyme. We'll just give it a name. Let's imagine that its name is EcoR1. So EcoR1, that's the name of one type of restriction enzyme. EcoR1 is actually able to recognize this palindromic sequence. So as EcoR1 is floating around the bacterial cell, it'll recognize this sequence, and if it's methylated, it won't touch it. But if it's unmethylated, as in the viral DNA, it will actually cleave it. So it'll come here and it'll cleave it. Let me actually use a different color. So it'll cleave the DNA, and it'll cut it like so. And what that'll do is it'll result in 2 strands, because it just cut the DNA. So now what we're going to have is we're going to have one strand that's going to be G, and then this bottom strand, TTAA, and then we're going to have this top part over here. So we're going to have an AA, TT, C, and then a G down here. So basically, now these 2 strands are just floating around, and these ends down here, so this end kind of hanging over the edge, they're known as sticky ends. So these are known as sticky ends. And the sticky ends basically will float around, and if they contact one another, since they're complementary strands, they'll just reanneal. So normally, if this gets cut, then you'll have 2 sticky ends, they'll float around, and then they'll reanneal. And we can actually take advantage of the fact that we can have the strands reanneal, and we'll talk about different ways that we can actually use.
Okay, so hopefully that kind of goes over a little bit more what I talked about in notes yesterday. So basically, I don't really care if you understand all that methylate stuff. That just tells you how it got its name. Okay, I'm more concerned about what are these sticky ends? They keep talking about these sticky ends and how they work. I think that's the tricky part is how does these restriction enzymes work? Well, what they do is they find this palindromic sequence and we cut it, and you saw that. Now the nice thing is when we say reanneal, that means they can go back together. So what we do is we just take a section, 2 strands, we split it into 2 strands. Now that we have this strand has a sticky end and this strand has a sticky end, we can take a gene from even another organism like a human insulin gene. We can go into a bacteria which has a circular DNA, and we can cut out that section, and then we can put in the code for insulin. So we're not changing the bacteria's DNA totally, we're just changing one part of it. Okay, so by inserting this gene in there, okay, we still have bacteria. Bacteria still is producing more bacteria, but they can do it so much faster. If we put the human insulin gene in there, now when they reproduce, they produce this human insulin that we can then take out and mass produce it, and it's cheaper, it's faster to produce, and people benefit from it. And people take it into their bodies that are diabetic because it is human insulin. So that's one way we found an advantage to doing this. And we'll look at some other things too, but that's basically what I was talking about yesterday. I hope that makes sense. So think of that gray part right here. That's just a different sequence that, and you can see it reannealed. So we're not really changing the DNA of the bacteria. We're just inserting what we want in there so that, and DNA ligase kind of seals it up. So we can just use this for, again, a useful purpose, correct? All right, so that's kind of the idea. All right, so that is basically how that works. And then this is just another picture. You can kind of see maybe a little bit more with that. I don't want to spend tons of time on this, but you can see same deal. There's my circular DNA. We've taken restriction enzyme, we've cut out the black part. We put the gene of interest, which can be anything. We insert it in there, maybe human insulin or whatever, and then we can start, that bacteria will start producing that, okay? That's good. It also can be used for bad, because what if we put something bad in that black spot, right? So, I mean, it's all about how are we using this technology? And like a lot of things in science, you guys all know this. We can use it for the good of mankind, or we can use it for bad, right? And that's what we have to be careful with. And you can see some of the uses we're looking at. Maybe a protein we can insert in there to dissolve blood clots, clean up oil spills. This is cool stuff right here. We've done it with crops for years. Don't get hung up and get caught up in the social media stuff. GMO crops are not all bad. I plant GMO corn and soybeans to help my uncle farm. And we put genes in there for pest resistance, for drought resistance. It's not going to contaminate, infect the food chain. It's not going to cause cancer and things like that. We're very selective. We're not changing the entire genetic code. We're just inserting a particular gene in there to make that crop or that food or whatever better, right? Human growth hormones, whatever. So lots of applications for this. Human insulin we talked about. So kind of keep that in mind, how this can be used. It can be used for good. It can be used for bad, right? So it just all depends on the application, okay? I mean, it all comes down to application, right? So that's basically what's going on with my computer here. So let's kind of wrap up notes, talking a little bit about some of the other things, some of the tools that help us do what I just showed you. PCR, gel electrophoresis that we did a lab on. There's lots of different things, lots of tools in a tool belt, the old saying goes. So just some terminology. Transformation is this when a bacteria takes up a gene of interest, which I just showed you. That's called transformation because we're transforming it into something else. PCR is kind of a interesting device. It's called polymerase chain reaction, and it can copy pieces of DNA without the use of cells. This is a wave of the future. A lot of the DNA that we used in our gel electrophoresis lab was made this way. What if we can take that same thing I just showed you, but without using bacteria and live things, can we take it and mass produce it in a machine? PCR can do that, okay? Which has got a lot of applications. Gel electrophoresis, I just showed you that. We're using a current, and we're pulling DNA through that gel, which has space in it, so we can separate. We use that in forensic science a lot. And DNA microassay is just studying the many genes at the same time, okay? So here's PCR. It's just basically taking a target sequence. We use a restriction enzyme. We cut it out. We denature, we anneal, we extend it. I'm not going to go into a lot of details with that. And then we can mass produce it, which has a lot of applications, like in human insulin, things like that, okay? So you can see why, we can have bacteria produce this, then we can take it out of the bacteria and mass produce it with the PCR machine and really benefit mankind, okay? And lastly, I just want to kind of finish up with gel electrophoresis, what we did. You should know this because we did our lab, but I don't know if you know that, but when I put that orange lid on there, that keeps things flowing one direction. So then we want it to flow from a cathode, a negative, to a positive. So that's why those bands pulled down through the gel. It's high voltage electricity. I still remember our old machines were not foolproof. We've come a long way with technology, and I still remember I had several groups. I had 10 gels in this great big electrophoresis machine. We put them in there, spent hours and hours getting them ready, and one of my students put the lid on backwards, and I flipped it on. I came back 30 minutes later, and all of them ran left instead of right, and all my DNA ran off into La La Land. So we ruined all those hours of work. So we try to keep this foolproof now, but basically we know we're taking advantage of the electrical charge on DNA, which is negative. And we know that if DNA has a negative charge, if I can hook up a positive charge to the other end, I can pull it that direction. And then we know that different DNA has different weights to it, different genes, and The heavy ones deposit early, the lighter ones go towards the bottom of the gel. We get this banding that can identify people, and that's what we did the lab on. All right. Hopefully that makes sense. I hope I'm not going too fast through all that. If you have questions, let me know. Have a good day, and we'll kind of pick up there next set of notes. Hey, have a good one. See you. Bye.